Now, students, we are going to discuss the properties of the enzyme. So, what are the enzymes? Enzymes are the protein catalysts that increase the velocity of the reaction. However, there are certain RNAs which also have ability to catalyze the reactions to show their catalytic activity, and such RNAs are known as the ribozymes that basically affect the phosphodiesterase and the peptide bonds. So, but these RNAs are less common than the traditional protein catalysts. Now, what are the properties? Firstly, we are going to discuss the active site. We have an enzyme that contains a special site, and this special site is formed by the folding of the protein. This site actually contains the amino acid side chain that interacts with the substrate. Whenever there is interaction of the substrate with the enzyme, there will be formation of the enzyme substrate complex. Now, this binding of the substrate with the enzyme will result in conformational changes. Thus, after that, there will be the formation of the enzyme product complex. This whole reaction is reversible. Ultimately, we will get enzyme and product from dissociation of this enzyme product complex. Now, what is the efficiency of the reaction? Enzymatic activities are catalyzed by the enzymes. So, enzyme catalyzes the reaction at very high velocity, that is from 10 raised to power 3 to 10 raised to power 8 times faster than the normal reaction that don't undergo catalyzation. So, now, what is the turnover number? The number of substrate molecules which are converted to product by the enzyme molecules per second. Don't forget per second. Don't forget the time. Is the turnover number. Turnover number is the number of the substrate converted to the product with the help of the enzyme within a second. So this turnover number is usually 10 raised to power 2 to 10 raised to power 4 per second. Now, what is the specificity? Enzymes act specifically for the particular reaction for the particular substrate. So enzymes are highly specific. They catalyze only one type of the chemical reaction. But everywhere we have exceptions. We have different enzymes that catalyze the same reaction, and such enzymes are actually the isozymes. Now, what are the haloenzymes? What are the ecoenzymes, cofactors, and the coenzyme? We will discuss these properties of the enzyme in our next lecture. So, other properties are regulation. Enzymes have ability to regulate the process in a very well-designed way. They have ability to increase the reaction according to the body requirement. Our body is designed to decrease the reactions according to the cell requirement. These enzymes are either present within the cell, they are either present in the plasma. We have seen that there are certain enzymes which are present within the organelles. For example, in the mitochondria for the TCA cycle, fatty acid oxidation and oxidation of pyruvate, we have certain enzyme. And for glycolysis, for the peptone phosphate and for the fatty acid synthesis, we have certain enzymes in the cytosols. Secondly, we have enzymes that are released by the cells in the plasma. We have enzymes in the plasma which are released as a result of the turnover of the cell as a result of destruction of the cell. So this was all about the properties of the drug. In the next lecture, we will discuss certain definitions which are associated with these enzymes.